Thanks for joining us today. It's my pleasure to introduce Robin Hopkins for our seminar today. Robin started as an undergrad at Brown University under Annie Schmidt, who instilled her with love for plants and evolutionary biology. She then went to Duke to do a PhD with Mark Rauscher. In Mark Rauscher's lab, Robin kind of carved her own space and ran with this idea of building a new system out of these beautiful, well, mostly pink, as you'll see, but she will insist in calling them red flowers and from the genus Phlox. She actually pulled this like tiny little thread from like old papers that spoke of a highly controversial thing called reinforcement. It's kind of the notion that natural selection will complete reproductive isolation because of like the crappiness of the hybrid. And actually, through her dissertation, we now have the most beautiful example of how reinforcement in nature actually does happen. So this genus Phlox has become the model system for reinforcement, and now thanks to the rest of her work at Harvard, where she ended up as a professor, is becoming a much larger system for the evolution of reproductive isolation, adaptation, mate choice, ecological traits in plants. As a lot of people have already said before me, including in beautiful features in Glam magazines, Robin is a person who goes soup to nuts, which is an expression that as a foreigner, I didn't really know. Apparently it means that you go from the beginning to the end, as in for a meal, of a story. So she starts from like the ecological side, does the natural history, and all the way down to even the nucleotide changes that might lead to adaptation. And maybe she'll tell us a little bit more about that. So as I said, after Duke, she did a short postdoc at the University of Texas, and then straight went to Harvard, where she got tenure and full professor last year. And that's it. So please.
Thank you all. I've had an amazing visit, thanks especially to Rafa. He forgot the whole bit that when I was a graduate student working in Texas on this system, he was the family I went to and taught me everything I know about math and coding and actually evolutionary biology. And so I am still grateful for every moment of collaboration and insights that I gained from him. And it is such a treat to be here in his new home, and which I guess is not really all that new for you, and get to meet everyone and hang out with everyone. I've had such an amazing time, so thank you. I am excited to share with you two vignettes, but first I'm going to start with a little bit of what motivates me, which is classic naturalist diversity slides. No, what it is like incredible amounts of biological diversity, and this in particular is really exciting to me because I like to pretend that all of this diversity evolved to make my gardens beautiful and my bouquets exquisite. It turns out it didn't, right? So all of this diversity evolved to optimize sexual reproduction. And sometimes we might consider that mate choice. And so what I've realized in my lab is that a ton of the research that's going on is really focused on these ideas of mate choice. And when I and many people think of mate choice, we think of this. We think of like colorful animals that squawk and dance and fight and, make funny smells or whatever. And that is really charismatic examples of mate choice, which are, traits that evolve to optimize or control reproduction. And I would argue that this is kind of the same thing. Right? That plants have evolved incredibly creative and amazing mechanisms to determine how to get the optimal mates and optimize reproduction. And so in particular, I think in plants, there's two really important decisions that have to be made with these organisms with their brains. And one of them is hybridization. There's a lot of pollen in the air. We all feel it when we're sneezing in hay fever season. But plants feel it all the time when pollen is hitting their stigma, and they have to figure out what plant to actually mate with. Is it the same species or a different species? The second really important mate choice that faces a lot of plants is selfing. So many plants will make both pollen and pistils, And so both of these make gametes, right? Classic sperm gametes in the pollen, and eggs or ovules in the pistils. And if a single plant makes both of these, they can self-fertilize and produce offspring with being both parents in that sexual reproduction. And you can imagine that sometimes that's really good. You have mate assurance. You don't have to worry about pollinators or wind or whatever getting your pollen to success, but sometimes that can be really bad because of depression. And so I would argue that these are two incredibly important mate choices that plants have to make and have evolved mechanisms to mate. And I think that the evolution of these mechanisms largely fall into two big categories, or are most charismatically known in two big categories. And one is the manipulation of pollinators, evolving traits that will either attract or dissuade or manipulate the way in which pollinators move in order to control which pollen arrives on the pistils. The second is a little bit more subtle, which we don't see quite as obviously, but that is pollen recognition, where there's incredible biochemical interactions between the pollen and the pistils in that process post-pollen landing on the pistil to seed formation. And so we're going to explore both of these arenas in much more detail, but we can imagine that this is actually, in many plants, quite a long path from pollen landing to making a seed. And these mate choices have, just like with animals, many implications for the success of plants and plant lineages. The most obvious of which is the health of the offspring, right? You optimize your mate, or plants optimize their mates, and that can really affect the actual fitness of their offspring, the ability to make seeds and the success of those seeds. It also has really big population genetic connotations or implications with the connectivity of populations and the amount of gene flow and spreads of adaptations or maladaptations. These mate choices also have broad phylogenetic patterns, right? We kind of classically know that pollinator manipulations and pollinator syndromes and such have huge implications for how diversification in angiosperms happens. So the ecological and evolutionary implications for these mate choices are really big, which has been sort of an exciting theme in the lab. I'm going to tell you two vignettes today. One of them is thinking about this mate choice of hybridization and how plants can manipulate pollinators to control rates of hybridization. And the second vignette is going to be about selfing or controlling self-fertilization and looking at these pollen pistil incompatibilities or interactions that can evolve. So in Act 1, I get to share with you this organism, this phlox. So this is Phlox stromundii. It is a Texas wildflower. It's an annual. It is pollinated by butterflies. It covers the roadsides. It's quite gorgeous. And what's really striking about this particular species is that it has these two flower colors. And this is where Rafa teases me all the time. Everyone does. Because we have this deep dark red color, but we also have this purple violet color that we call light blue. No, you are not blind. It is purple, but we're calling it light blue because it looks prettier on these pictures. Okay, so the distribution of color of the species, it's really soggy and wet out there. We're dealing. All right, so the distribution of these two colors of the species are such that most of the range is this sort of light blue, purple, violet color, which looks like every other phlox flower you've ever seen that is natural. But then in the eastern edge, there's these bright red populations. And what was observed long ago in this paper is that these areas of dark red flower colors coincide with an area of sympatry or geographic overlap with this closely related species, Phlox cuspidata. So these are super closely related, and you'll notice they're the same color, this light blue, purple, violet color. And so it was hypothesized that this flower color in sympatry evolved to diverge in order to manipulate pollinators and stop hybridization. And this process is, as Raphael alluded to, this process of reinforcement. And it looks something like this. You've got two species, say some two blue colors, say they share pollinators, and they hybridize. Those hybrids are bad, sad flowers. If there's selection against these hybrids for whatever reason, that can then favor the evolution of any trait that would change the pollinators or stop the hybridization and such that there's no more hybrids and only pure species are formed. So this process of reinforcement is selection to stop hybridization, right? And it was thought that maybe this red flower color did just that. So for this first vignette, we're going to answer two questions. Is there plant pollinator mate choice in this system through this flower color? How is it evolving? Do we have evidence to understand why these genetic mutations have occurred? And then finally, what are the actual genetic mutations that are underlying this really important evolutionary phenomenon? So this starts with the pollinators, where this phenomenal graduate student, Grace Bergen, went out in the field and spent hours and hours and hours watching natural populations of flocks, both red and blue and all these colors. And after about 3,000 visits throughout the whole day, morning to night, 90% of the visits are by a single species of butterfly. And it's this guy, this Batis philonor, which is a giant, they live around here too, these giant black with purple iridescent butterflies that are incredibly effective and incredibly abundant in pollinating Phlox drummondii. Turns out they also pollinate Phlox cuspidata. And so we very quickly we manipulate pollinators to, does Phlox manipulate Batis philonor, right? This one pipevine swallowtail butterfly. So to look specifically at this question of how does this change in color manipulate the behavior of these butterflies, we use controlled potted plant arrays that look exactly like this, where we have pots of plants that are abundantly blooming. We put them out in these arrays where we can control the number of flowers and species and very controlled. I sit with my giant sun hat and watch the butterflies move around and ask, do they move differently between species of the same color versus different color? And sure enough, when I look at these arrays that include this dark red and light blue and this other species, I see that a butterfly that's on a light blue drummondii plant, about 30–40% of the time, the next plant it goes to is a cuspidata. This is moving pollen to create a hybrid. Now I compare this to a butterfly that is on a red plant, and only about 10–15% of the time does that pollinator then move to a cuspidata plant. So pretty quickly we see that these pollinators behave differently when they're on a light blue versus a dark red drummondii. And the same is true in the other direction, that a pollinator on this cuspidata moves much more frequently to a blue drummondii than this dark red drummondii. So just through this work, we can say, yeah, seems to be pollinators are moving differently. That's pretty cool. This is a mate choice or the evolution of a trait that controls and manipulates the way that mating is happening in this system. How is it evolving explicitly? Is it reinforcement? Is there hybridization? Is that hybridization costly? And do we actually see variation in this hybridization like we predict from this scenario? So this work is published, so I'm going to kind of move through it pretty quickly, but I will tell you they hybridize. So here's a hybrid. Here's a drummondii. Here's a cuspidata. The hybrids are there. We've also done a bunch of genomics, and we can see in populations that we can use structure where each individual is a little column and the color represents the inferred proportion of each genetic background. And there's a lot of drummondii, and there's a lot of cuspidata, and then there's clearly some hybrids, which look like F1s or back crosses. So there are current hybrids. We also can use population genomic data to infer historic hybridization, and sure enough, we can infer that there's been historic signals of ancient integration on the order of 3 to 7%, depending on where you are in the range and what it is. So yes, hybridization happens. Is hybridization costly? So this was some work that we were really trying to nail down with a postdoc who was in the lab a few years ago, Sevan Suni, where she did a whole bunch of crosses where we look at cuspidata plants, which we can cross within other cuspidata plants. And this is really depressing. Every cross gives you like 1 to 3 seeds. It's pretty anticlimactic. And then the same with drummondii, you get a few seeds, right? And then she could make hybrids. Turns out you can make hybrids pretty well. And then she crossed those hybrids back to the parents and to themselves. Yeah, they don't make seeds. They're really, really strong hybrid sterility, both on the male side and the female side, implying that you can eke out a few seeds here and there, but it looks like these hybrids are evolutionary dead ends. So when these hybrids are formed in nature, which we see, there's a huge cost to that. It's basically a dead offspring with a little bit of leaking through. So then the question is, can we actually link this change in pollinator behavior to a change in hybridization rate that will affect then the evolutionary course of this species? So we went on the field and we planted up a whole bunch of cuspidata and a whole bunch of light blue drummondii and a whole bunch of dark red drummondii. And we let the pollinators come, these Batis come and pollinate away and set a whole bunch of seeds. We then collected those seeds, grew them up in the greenhouse and did paternity testing and asked, is cuspidata your dad or is drummondii your dad? Are you a hybrid or are you not? And in this way, we can determine that about 40% of the offspring of drummondii were actually hybrids when that drummondii was light blue. whereas only about 20% were hybrids when that Drummondii is this dark red. So again, we see this 50% reduction in hybridization when we evolve different colors. That seems pretty big, pretty exciting. So yes, there's hybridization. Yes, it's costly. Yes, there's variation in hybridization depending on flower color. We can conclude, indeed, the system is evolving through reinforcement. Okay, but I feel like I'm in a room of geneticists mostly, right? So how is that doing it, right? What is the actual genetic mechanism that is allowing this mutation to occur? So for that, I'm going to tell you a little bit about some old classic genetics that we merge with some genomics and functional genetics to get down into what is the genetic mechanism of this change. First, I'm going to tell you that there's more colors than just two. When you cross these, they can recombine to create 4 flower colors. And in those 4 flower colors, we see that one locus or one change corresponds to the intensity of the flower color. That is the amount of pigment made, with these guys being light and these guys being dark. And some candidate gene associations looks like it involves something about this transcription factor, R2-R3-MIB, which is known in many systems to affect the amount of anthocyanin production in species. The second change is a locus that controls the hue or the type of pigments that are produced. Here we go, ready? These make blue pigments and these don't. Now you know why I call them blue. So these make both blue and red pigments, and these make just red pigments. And that change is caused by a single locus, and that is associated with this branching enzyme in the anthocyanin pathway, F3'5'H. So for the remainder of the talk, we're just going to talk about this intensity locus, and it's because this phenomenal graduate student, Austin Garner, in collaboration with a postdoc, Andrea Berardi, and Andrew Cameron, a technician in the lab, really wanted to dive deep into this particular gene and understand what are the genetic basis of that. And so the first thing that they did was this time course of expression, right? Like, what do we see as the expression profile of this gene throughout development? So we take flower buds, which is where this gene will be expressed in order to make pigments in the flowers, way early before we see any color, to open to 3 days open when it starts to wilt. And sure enough, we see peak expression of this transcription factor right around the first day of opening, a burst of expression corresponding to this open flowers and dark pigmentation or pigmentation. So this is for the light individual. They then looked at dark individuals and they see a totally different pattern. What they see here is that we have really early expression of this transcription factor that is significantly higher than what we see in the light individuals. But actually, by the time we get to peak expression in the light individuals, they even out and are the same. So we hypothesized pretty quickly that this change in color is due to some gain of expression signaling early in development that allows the put down or laying down of greater pigmentation. So to confirm that it is this transcript abundance or gene expression that is responsible or is sufficient to change flower color, we, as in they, developed this gene knockdown protocol in our plants that's called VIGS, or viral induced gene silencing, where basically we trick the plant using a virus to use sort of an RNAi mechanism to knock down the gene of interest for us. So we basically infect it with this agrobacterium that has this virus that contains a plasmid that has our gene, a chunk of our gene of interest that triggers the immune system, knock down it. So anything infected is going to have a knockdown of the transcript numbers of this gene of interest. So we do it with our MIB. We degrade all of our MIB transcripts. And the beauty of this is that you get a mosaic infection, where some of our plants, the parts of branches of the plants remain dark and unsilenced. So this is what the plant looked like before we infected it. And in those branches that are not infected and silenced, there's super dark flowers. We then, after infecting, some of the branches are super light flowers. And we show that indeed, this has almost no transcripts of this R2-R3-MIB that is our candidate gene, and it looks really similar to the natural light flower color. And using quantifying flower color, we can show that it indeed is not statistically different than the natural light flower colors. So we can do this for both light and dark, and you can see, hopefully, the mosaic patterns that we get both within flowers, but also just when you get a good knockdown, you look exactly like the light. So this gave us a lot of confidence that we have the right gene and that we're talking about gene expression. There's something about the signaling of expression of this gene that is causing this change in flower color. All right, so here's the flux. example of a piece of a flux genome, right? Where we might imagine that there's the gene that's sitting in there, and there's something in this area that is controlling expression of this gene. So our goal was to go to this region of Texas where the red and the blue overlap and freely recombine with each other, and the dark and the light freely recombine. You get these mixed populations where nature can do all of the genetic process for us. And then we can use an association study and see if there's any site that is associated with the light and the dark. Well, the problem with this little scenario is that our genome is giant. So Lox has a 6 gigabase genome that's about twice as, well, it is bigger than twice the size of the human genome. And so it's a bit of looking for a needle in the haystack. So this graduate student, Austin Gardner, found this really cool technique done by Samplex, which is like an extra nanopore sequencing at a targeted area. And this allowed us to sequence about 80,000 base pairs just around our gene of interest that we target. So we can do our association not on the 6 gigabase genome, but on 80,000 base pairs right around the gene we're interested in. So this is what it looks like. We get these long read nanopore sequences at depths of, between 100 and 500 X coverage in about 80,000 base pairs right around our gene of interest. So it's beautiful coverage, which allows us to get SNP variants and structural variants all in this region in particular. So we do this for about 120 individuals from natural populations that show natural admixture. We sequence them, we do a simple association to ask which variants are associated with this change in flower color from light to dark. Are you guys ready for our Manhattan plot?
Yes.
Seattle in the fog, baby. There is nothing, there's one SNP, maybe this one too. This SNP alone explains 70% of the variation in phenotype when we quantify it using a spectrophotometer. It is perfectly associated such that dark individuals have 1 or 2 of the dark allele T, and light individuals only contain the G. I was lucky. So that was really exciting. Using this targeted approach and allowing natural recombination to break up everything else that would have been in LD with this causal mutation, we are now able to find the mutation that changes expression in this gene in dark flower colors. So we zoom in and it turns out it's about 123 base pairs away from the start codon, the ATG. And it's sitting right in the primary promoter region in the middle of these cat and tata boxes. And what it looks like is in the light allele, you've got these couple cat and tata boxes. And in the dark allele, I don't know if you can see this, but it is inferred to introduce both another cat box as well as an inverted cat box, as well as this E2FBF motif, which has been associated with attracting transcription factors that increase expression of genes associated with it. And it turns out this motif is actually responsible for another example in roses of upregulation of a MIB causing dark flower color. So this was super exciting. It looks like there's a plausible way in which this single mutation can change the expression pattern of this transcription factor, increase gene expression, and cause this dark flower color. It's a little bit of anticlimactic to be like, oh yeah, it's a point mutation. That was fun. But it's actually really exciting to think that this huge 50% reduction in hybridization can be a simple G to T mutation. Okay, so that first vignette, hopefully, was exciting. To me, it's really exciting because, like Rafa implied, I love being able to go out in nature and observe a pattern that seems to have biological significance, and then dig deep into the process of understanding what are the evolutionary forces and ecological interactions that are sort of underlying or affected by that pattern. But then really, I want to know the mechanism, right? Like, how do we then dig into these non-model systems and figure out what did evolution work on and how did we understand that mechanism? So for me, this is a really fun example of being able to go soup to nuts, I love that, from pattern to process to mechanism to understand how these adaptations can arise. Okay, back to selfing. Should we get an intermission? Canapes and cocktails in the lobby now. All right, selfing. We're gonna move on now to second story, which is about selfing and these pollen pistil interactions. So, probably not a big plant biology crowd here, but we're gonna bring you into the inner workings of a plant. Plant pollination happens. Pollen lands on the stigma, it grows a tube down the style, and then fertilizes doubly, fertilizes the ovule, and if it's successful and good, it will set a seed. And that continues on to the next generation. The problem is that sometimes this happens and those seeds are bad. Like often with self-pollen, because they're all homozygous and no good. And so many plants have evolved these mechanisms to reject the pollen before it can make a seed. So this process of growing these, of binding to the stigmatic surface, growing a polytube through the style, finding and then fertilizing the ovule, it involves many complicated biochemical interactions, all of which can go wrong or be manipulated to reject pollen. The most common of which, oops, oh, we're gonna have to go through this. Jumpy thumbs. Okay, most common of which, all just to show you self-incompatibility. So self-incompatibility is a genetic mechanism in which the pistil can recognize self-pollen and trigger the rejection of that pollen. And it happens in many different ways across many different plants. And what we wanted to know is, in phlox, do we see this pollen pistil make choice through rejection of self-pollen? How is it evolving? And then again, of course, what is the mechanism that is allowing for this variation or rejection? So the first thing we did was confirm that there is mate choice at this pollen pistil level in Phlox drummondii. So that was this poor postdoc, I don't have a picture of Federico, I'm sorry, Federico, who did hundreds and hundreds of crosses from drummondii individuals from across the range and looked at their seed set. And we would imagine that if it's self-incompatible, you get 0 seeds most of the time. And if it's self-compatible, you get 3 seeds max most of the time. So what did he find? What he found was sort of in the middle. There certainly are a lot of individuals that are entirely self-incompatible and set no seeds. But he found a fair amount, about 40% of individuals can set reliably at least 1, 2, sometimes even 3 seeds when you give it self-pollen. So we got pretty excited at thinking about what is this variation. The first thing we did was say, well, what is this variation? This is a phlox pistil. So the pistil looks like this. There's 3 stigmatic lobes. Each of these dots is a pollen grain that has germinated a pollen tube and it's growing down, and somewhere down here is an ovule that it's delivering sperm to. So this is what a compatible cross looks like. It is a party of pollen tubes, right? A lot of pollen tubes. We can zoom in. This is totally gratuitous. Aren't these awesome?
Yeah.
They're so cool. Pollen is just amazing. And then we can actually see it germinating right there. That's going to go into the papilia and down into the style and grow pollen tube. It's just really cool. Okay, here's the problem. When you have a self-incompatible cross or an incompatible cross, this is what you see. Sad stigma, actually it's probably a happy stigma, it rejected all the pollen. But every once in a while you could see like a little glowing dot. That's like maybe a pollen grain or something. You don't see any pollen grains, you don't see any pollen tubes. Nothing is bound to the stigmatic surface and germinated a pollen tube. So we're like, all right, this seed set thing is pretty clearly something happening between the pistil and the pollen. What we then wanted to know is this genetic variation. There's a lot of hand waving in the self-incompatibility literature that like, oh, it's a little leaky. Is this just like environmental leaky? Or are we talking about a trait that can evolve, that has genetic variation? So we went really old school geneticists on this, which was super fun, and did a selection experiment, right? If it is genetic, then there should be heritable genetic variation that will respond to selection. So, side effect of this really complicated experiment that we did, we ended up, this is another way to look at the distribution of a starting population, the density of seed sets or selfing, where these guys are self-incompatible, and this is sort of the proportion of the population that's self-compatible in the first generation. We then took this distribution to go on to the next generation in a classic artificial selection experiment. We then grew up those offspring from these controlled crosses and said, well, what is your compatibility? And sure enough, we shift the mean, estimate a heritability of about 40%. So this was a lot of work to say, yep, there's some genetic variation, that at least in part, this variation in self-compatibility, where some individuals are self-compatible and some are incompatible, has a genetic and heritable basis. So we got a yes. There is pollen pistil mate choice. Many individuals are self-incompatible. This is a genetic system that has genetic variation in the degree to which you are self-incompatible. So how is it evolving? Okay, to get to this, I need to back up and tell you a little bit about what we know about self-incompatibility systems. The first thing we know, so in self-incompatibility at the genetic level is often thought to be caused and is been caused in the systems that we look at by a multi-gene locus called the S locus. And this S locus, sure enough, has at least two genes that do not recombine, one that's expressed in the pollen and one that's expressed in the pistil. And if the pollen genotype matches the pistil genotype, then it recognizes it, triggers a self-incompatibility response, and rejects that pollen. If, on the other hand, the pollen that lands on that pistil is a different genotype, then it will germinate, grow pollen tube, and fertilize it. Okay, so it's a single locus, but a multi-gene locus that has evolved. Now, the thing about this S locus is that they have evolved, we think, about 15 times independently. So when we say S locus, we mean it's a locus that has two genes, one in the pollen and one in the pistil, but in those 15 times, we know that those genes are different, right? So it's independently evolved with entirely different genetic mechanisms. And only evolved about 15 times. We also know that it's been lost hundreds of times. So we look across clades of plants, and we know that some of them have incompatibility systems and some of them don't. And what we know is that the evolutionary direction is a loss of self-compatibility. They all started with it and then they lose it. It doesn't regain, plants don't evolve new incompatibility systems very often. We also know when you look across phylogenies that many plants that have been studied that are now self-compatible, what we know is that this is often associated with a broken S locus. That plants are either very, very good at being self-incompatible, or they're completely self-compatible and they broke their S locus. So that's sort of the general patterns in this system. So what we are seeing is that there's genetic variation within a species in the degree to which you're self-compatible and incompatible. So we wanted to ask these basic questions. Is this polygenic and actually like a quantitative trait? And is it associated with this S locus breaking? Like we see when we look across phylogenies at different species. Do we see that same kind of variation when we look within a species as it is evolving across populations? So we mapped it. We took a self-incompatible plant, a self-compatible plant. We created an offspring. We can trick these guys into selfing. You basically do bud crosses before anything is mature. You like trick it into mating and then it makes seeds, makes a whole bunch of seeds. And we quantify in those offspring, there's a bunch of individuals that are self-incompatible, a bunch that are self-compatible. We genotype them all. We did this 3 times to look for an association, 3 different mapping populations to look for an association between phenotype and genotype. In one of these, we genotyped everybody and did a classic quantitative trait locus experiment. In the other 2, we did a bulk segregant approach where we just categorized all of these together and genotyped them as one pool, and all of these together and looked at differences in allele frequency. In the end, we end up with 3 mapping populations where we can look across the 7 chromosomes of flocks and ask which regions of the genome are associated with more or less self-compatibility. So in the first cross, this was one of the bulk segregant crosses, so you have a G prime statistic, and we see one big peak that explained almost all of the variation in the variation in self-incompatibility. So we're like, oh, well, I guess it's like a gene of major effect on chromosome 2. And our second cross, okay, we got 3 peaks that explain some of the variation in the trait and none of them at the end of chromosome 2. So we look at our third cross and we've got 1 peak that mostly kind of explains some of the variation, and it's not in chromosome 2 or in chromosome 4 or in chromosome 5. So my graduate student just curled up in a hole and was like, I don't want to talk to you ever again. But I think this is really fascinating. And it is very true to the biology of this system, where we knew from the selection line experiments that you can take perfectly self-compatible plants, cross them to each other, and get an incompatible plant, right? This implies that there are many different loci that are causing self-compatibility. They're probably mostly recessive, and they complement each other in different ways, right? And so, yeah, any given cross you happen to unluckily do, you're gonna get a different locus. that is responsible for what is self-compatibility. So, I'm still tearing her off the ground and telling her life is okay, but I think what we can conclude is that, yeah, self-compatibility is polygenic and that there are many genes that can cause it, but sometimes a single locus is sufficient to break it quickly and you're done, which is complication of the answer. So are any of these, or all of these, hopefully any of these, actually the S locus, right? We now have 5 regions of the genome that are associated with self-compatibility. Hopefully not all of them are, but are any one of them something to do with breaking the S locus? So for that, we did some S locus mapping. Here we go. This is painful as I think, fun as a geneticist too. Okay, we have an individual. We assume it has to be heterozygous because it's self-incompatible, so it has to segregate alleles that are different. If we force it to self, it'll make 3 genotypes: homozygous S1, homozygous S2, and heterozygous. And our goal is to now group offspring that are full siblings into these genotype categories. And we do this by crossing them to each other. So we know any individual that's S1, S1 will be entirely incompatible with another individual that is S1, S1, that's its sibling. So you take siblings and you cross them to each other, and if they're incompatible in both directions, they are both S1, S1, or you cross them to each other and they're incompatible with each other, they're S2, S2, but they're compatible with the S1, S1. So using these patterns of compatibility across these giant mapping populations, we can then divide them into genotype classes of S1, S1, S2, S2, and again, use a bulk segregant approach. There they are, categorized. Bulk segregant approach to sequence each group and then look for allelic differences, hopefully 1. So again, our 7 chromosomes, our statistic. There it is. Thank goodness. This time it was 1. We did it multiple times, and it was always here at the end of chromosome 2. So that is where our S locus resides. And if you recall, it does correspond beautifully with one of these loci that corresponded to self-compatibility. So in this cross, it does look like a broken S locus will cause self-compatibility, but not always. So we conclude, yes, sometimes. Those are my favorite answers in biology, right? Like maybe, kind of, depends. Anyway, all right. But what's the genetic mechanism, right? What is the S locus? And how do you break it to cause self-compatibility? Well, how do you make it to cause self-incompatibility? So if we go back to this peak on chromosome 2, we know somewhere in here there is a gene expressed in the pollen, and perfectly linked is a gene expressed in the pistil. So we have an assembled genome, we have a transcriptome, we can annotate that genome, and it turns out there's 633 genes under that peak. It's not a lot, but enough that I didn't want to make a graduate student go through all of them. So the first thing we did was go back to understanding that this thing has evolved 15 times. And 4 of these different evolutions of self-incompatibility using an S locus system like this have been highly characterized. So in these 4 systems, we know the genes, okay, they're letters, right? We know the letters of the genes that are associated with self-incompatibility, and we can look in the transcriptomes from pistils and ask, are these genes there and are they expressed? So the first thing we did was look at a phylogeny, highly reduced phylogeny of angiosperms. All you need to know is that Phlox is here, and these are some of the other species, and they seem pretty-ish closely related, at least the closest related, nested inside this group that makes these sRNases, like the solanaceous plants. So the first thing we did was look for these sRNases, right? Using a Canada gene approach, could it be this? Long and short of it, no, they are not. It is an entirely different system. They make no sRNases in the pistil or anywhere else that we could find, and none of these other genes sort of popped up as being highly expressed, homologous to and highly expressed. Okay, so now we have a new system, but we're not at a dead end. We have 633 genes to choose from. And although there are none of these genes, we can learn something from these systems. And what do we learn from these systems? The first, we have very specific expectations about what an S locus should look like. The first thing is that it should have really high expression in the pistil. For all of these systems, it is like the #1 expressed gene or top 10 expressed genes in the pistil. And it's usually uniquely expressed in the pistil. So it's not highly expressed anywhere else. So that's a pretty good thing to know. The second is that they are highly polymorphic. So these loci are under really strong frequency-dependent selection. A rare allele will be compatible with everyone else in the population, and therefore rise in frequency because it is favored and it is compatible. Whereas a common allele is incompatible with anyone else who holds that allele and will therefore be lowered in frequency. So this system perpetuates a whole lot of variation, all of it at really high frequency. So we expect to see a really, really highly polymorphic locus. Also from other work in other systems, we expect that these systems are old and that these polymorphisms can be favored to be maintained across species boundaries. So related species, even distantly related species, can share alleles at these S loci that may be 10, 20 million years diverged, and the species are 1 million years diverged, right? So you get these really old alleles that are maintained across species that are closely related. So this work was done to narrow down by this phenomenal graduate student, Grace Bergen. I mentioned before was loving the butterflies and is now digging deep into the S locus work. And she wanted to go through each of these expectations and see if she could narrow it down. So the first thing she did was look at gene expression. So what I'm showing you here are some of the top expressed genes in the pistil. We do RNA-seq analysis on just pistil tissue, and she identifies a whole bunch of highly expressed genes shown in very, very dark purple. She then looks at the expression, compares the expression in these pistils to other tissues, anthers, leaves, pollen, and stem. And we pretty quickly see 2 genes that stand out, right? So these genes are really highly expressed in the pistil and not expressed barely at all in any other tissue. She then, using transcriptomes from a number of different individuals, she's able to make gene trees of all of these genes and look at polymorphisms. So basically what we're looking at is alleles that are really different from each other will be isolated by really long branch lengths, and alleles that are really similar to each other will have really short branch lengths. And so we can use these gene trees and the size and length of the branches in the gene trees to better understand which is the more polymorphic one. So we look here, different alleles are related with different base pair differences, and we look at a whole bunch of them, and there's one that has over 10 times the variation of anything else in this region and is hugely variable. Like, well, that's a good candidate. What if we look at other closely related species? So in this case, she takes 2 really closely related species and then another more distantly related species. She sequences this gene and a couple others from these species, pulls it out of their transcriptomes, makes gene trees of a number of different individuals with a number of different genes and then across different species and looks to see. So this is our gene 52568. And what all you need to know is that the different colored dots are alleles from different species and they don't group together. And if you look at the gene right next door, all the colored dots group together. And the other gene next door, oh boy, forward clicking. The other gene next door, right, all of them group together. And so what this tells us is that this gene 52568, different species are still harboring the same alleles that are really distantly related relative to the relationship of the species. So we got really excited. That this is like a really good candidate for the pistil side of our S locus gene. So she went back to this cool technique that they developed in the lab to do this gene silencing. And she hypothesized if I take a self-incompatible plant and I silence the pistil gene at the S locus, it should become self-compatible, right? So let's do it. Here's the trick. So again, we make a plasmid, then we infect the plant, it degrades it. The problem is, again, that this gene silencing is a mosaic. And you can't see the S locus. So she also used this little trick where she put a flower color gene on the same plasmid. So now we can see it. So this is a red plant, and where we infected it and silenced both the S locus and, or the candidate gene 52568 and the flower color gene, we now have white flowers. So we have a visual marker that says silencing is successful on this branch, in this flower, in this way. And then she takes silenced and unsilenced flowers and she asks, are you self-incompatible? So we start with, again, just the flower color gene. There's no S locus here. And we take unsilenced flowers and silenced flowers, and as a control, they don't set seed at all. They're like, guess what? Flower color doesn't control self-incompatibility. Surprise, surprise. Okay. Then she takes an individual that is homozygous. We tricked it into being homozygous for this S4 alleles. It's just the fourth allele we sequenced. The S4 allele was nicely put on, targeted for silencing. And we asked, how do we compare unsilenced, should be self-incompatible, to silenced? Boom. It was amazing. She did controlled crosses and she got seed. She then left these plants. They were infected, they were silenced, and they just started setting self-seed everywhere. It was crazy. It was so effective. Guess what? Causing self-compatibility. Okay, so then she's like, all right, did I just break the self-incompatibility system? Or can I determine that this is specifically a self-recognition locus, right? Not just the rejection locus, but the self-recognition. That's okay. So she took a heterozygous plant. This one is an S2, S4. Now these alleles are super highly diverged, like pain in the neck to PCR and amplify anything but a single allele at a time. You need allele-specific primers to amplify them. So we were pretty hopeful that indeed, if we just target the S4 allele, we'll knock down the S4 allele, but keep the S2 allele expressed. And we can show we can do that. So it's a heterozygous plant with the S4 allele knocked down and the S2 allele functional. What we would predict is if this gene is indeed identity responsive, it should be able to recognize the S2 allele, because that's fully functional, but it should not be able to recognize the S4 allele. So when we put S4 pollen on it, sure enough, it doesn't recognize. Now when we put S2 pollen on it, it does. And so she can show that the knockdown was allele specific and that the effect on self-compatibility was only specific to the allele that she knocked down. That there's nothing about, that the rest of the pathway is fully functional. It's only the specificity that is knocked down. So this is incredibly exciting. In the process of this, she also found, through sequencing and doing crosses, a self-compatible allele. So one of these, basically the gene under that peak, the allele under the peak that is a broken S locus. So we're really excited about digging into that and figuring out how did it break and what is broken about it, but they're kind of a pain in the neck, so we haven't gotten very far. Okay, so what am I going here? Oh, I wanted to know what it was. What is it? 52568, that's what we were gonna ask, right? We know that it's 52568, has high expression, it's highly polymorphic, there's trans-species polymorphism. We can knock it down and show this allele specific. So what is it? Okay, turns out it is a G-type lectin receptor tyrosine kinase, which, I don't know, does that mean anything to anybody?
Yeah.
Here's what it means. Good. It's a really cool one once you learn about it, right? But I'm like an evolution bot, right? So, okay, it's got this single transmembrane domain, and the extracellular domain is the highly, highly, highly polymorphic part, which makes sense. It's what's receiving the signal and matching with the pollen. And then there's these signaling domains that clearly can transmit this signal of this is self-reject, reject, reject. So it makes a whole lot of sense, and it's really exciting. Particularly because if we step back and we look at all these other systems, again, here's FLOPS. It's nested within these RNases. This guy in brassicas are really different than the sRNases and anything else around it. And it turns out it is also a tyrosine kinase. And is different and independently evolved, but it's in the same gene family and has all the same domains and functions as the gene we found in FLOPS. And so we're super excited at thinking about this convergence. Prior to this, we'd only had 4 systems and they were all really different. And finally, we have another system that looks like it's the same and maybe convergent in at least the type of molecular mechanism it's using, co-opting to have this self-recognition. So we're super excited about digging in to this, finding what the pollen gene is, and figuring out how much of the rest of the pathway is also convergent with what we know in brassicas. All right, so what is the genetic mechanism? Transmembrane receptor tyrosine kinase, a mouthful. But importantly, I think, again, we have this beautiful pattern of variation and self-compatibility within Phloxtra and a deep dive tells us a little bit about how it's evolving. Is it evolving in the same way we see these traits evolving across big phylogenetic clades, or is it evolving sort of differently within the species? And what it looks like is a little bit of both, and it's going to be complicated to untangle why and how it's evolving within the species. We then can do some deep dives into the mechanisms by pulling out what is this self-incompatibility locus and starting to look at why it might function the way it functions, but also break and therefore cause self-compatibility. So we're super excited about that. All right, so maybe you've seen a theme here that what it really excites me about these questions is this integration where we can get these amazing patterns in nature that really inspire us to think about the phenomenal evolutionary processes that are going on around us, but then getting into the like, how does it do that is really exciting. And for me, it allows me to step back and look at this variation. Okay, maybe it didn't evolve for my garden, but now I can think a little bit more clearly about why it evolved and how it evolved. How did we get this variation that is so extreme? And so with that, I give a giant thanks to my amazing lab. They did all of this development of the tools and the resources and also the amazing science. And of course, my funding, and we should have some minutes left for me to take questions. So thank you.